Hello friends, welcome in Pharma Talks by Arpit Pandey, and the today's topic is analytical method validation. Here we are going to know what is the analytical method validation, why it is important, what is the need of the analytical method validation, and why we perform it during the method development and all. Okay, so now let's get started. What is analytical method validation? Okay friends, why method validation is important, so the purpose of analytical measurement is to get consistent, reliable and accurate data. Inaccurate measurement result can lead to tremendous costs. Equal importance of those working in a regulated and in an aggregated environment, US FDA, EMEA, EPA, AOAC, ISO, TGA, they require the analytical method validation data. Background: NDA ANDA must include the analytical procedure necessary to ensure identity, strength, quality, purity and potency of the drug substance and drug product. And the details about this you can found on the 21 CFR 314.550 D1 and the 314.94 A91 topic. Data to establish and reliability. It is the background of the validation. Validation is an old concept, but there are many problems. What are the problems we face during the analytical validation? Lack of documented procedure and documented validation result. Sampling or sample preparation steps contribute to overall error. Accessories and material used for equipment qualification are not qualified. Limit for operational qualification. Lack of software validation and computer system validation. Qualification and validation are done at just one particular point in time. Adaptation of acceptance criteria for qualification of new system. So validation activity include the complete analytical procedure. When you are going to perform the validation, it include the complete analytical procedure. It start from sampling, sample process preparation, analysis, data evaluation, reporting. All is this include in the analytical validation procedure. So what thing you have to consider prior to start the method validation? Something you need to make or adjust the resources prior to start the method validation. The first thing is the suitability of instrument. How you can ensure status of qualification and calibration can ensure the suitability of instrument. Suitability of material, that is status of reference material, reagent, placebo, lots, etc. Either it is not expiry and the COA is available. All these you have to verify prior start to validation. Suitability of analyst. The analyst who perform the validation first ensure that they have the analyst have the analyst qualification record, training record about the instrument, software and the technique which he is going to validate the method. Suitability of document, written analytical procedure and prior approved protocol with pre-established acceptance criteria. It means that when you are going to start the method validation, it is suitability of document must ensure prior to start the validation. So what thing you have to ensure during the suitability of document? The first thing is you have the written analytical procedure, means you have the proper STP, you have the proper approved validation protocol and all with the acceptance criteria. After that, if you prior consider these and you have all these resources, then you are going to start your method validation activity. So first thing you have to ensure all these points. Validation steps. What steps have you taken during the validations? Define the application, purpose and scope of the method. Means what is the application of your method, what is the purpose of your method and the scope of the method. What is analyte, what is concentration and sample matrix. Develop analytical method. Develop a validation protocol, qualification of instrument, qualify or trained operator. Qualification of material also need to ensure perform free validation experiment. Like during the method development part, you have to perform some mini validation experiment like accuracy, specificity, linearity, LOD, LOQ precision, which can more help during the validation of the method. Adjust method parameter and or acceptance criteria if necessary. Perform full validation experiment. Once you make all these things, then you perform the full validation experiment. Develop SOP for executing the method routine analysis. Document validation experiment and result in the validation report. So this graph shows that the whole analytical validation procedure in a simple step. What you need to prior consider during before the validation, what things you have to justify. First you have a sample, then the first thing you have to system suitability things. So if you have a sample and analyst and a method, you have a sample, you have a method, and you have the analyst. If you have a method, you have to just verify the system suitability criteria, and the analyst who perform the validation first verify the calibration and the qualification status of the instrument before starting or proceeding the validation. After that you should have to perform the validation activity. So the first question arises in our mind, what is the verification and what is the analytical method verification and what is the analytical method validation? There is a little bit difference, and some more of the persons are confused about this. So now we are going to explain it, what is the verification and what is the validation. Okay friends, so any compendial or non-compendial method, okay, any compendial method we need to perform the analytical method validation. What is the compendial method? Any regulatory analytical procedure which is given in the USP, NF, EP, IP. So it is the compendial method which is given in the pharmacopoeia. So if you need to educate or take the method which is given in the pharmacopoeia as it is in your written procedure or standard testing procedure, you have to only perform the analytical method verification, not the validation. If you develop a method which is the non-compendial, you develop the method in-house, so you have to perform the complete method validation as per the validation guideline given in the ICH or any other regulatory agencies. So it is clear that any compendial method you have to perform the analytical method validation, and if you have a non-compendial method you have to perform the validation. Regulation and guideline of validation. Which guidelines and what guidelines give the data and give the guideline, give the necessary regulation for the validation. So US FDA 21 CFR and Code of Federal Regulation Part 210 and 211 gives the in Part 210 CGMP in manufacturing, processing, packaging or holding of drugs journal. In Part 210 21 CFR Part 210 gives the CGMP current good manufacturing practices in manufacturing, processing, packaging, holding of a drug journal. In Part 211 CGMP for manufacturing practices for finished pharmaceuticals. Which guideline in ICS give the steps about the validation? Q2A text of validation of analytical procedure and Q2B validation of analytical procedure methodology. If you talk about US journal chapter, so chapter 1225 give the validation of the compendial method. We here in the USP journal chapter 1225 you can find the all detail of the validation of the compendial method. The accuracy, sensitivity, specificity, reproducibility of test method employed by the firm shall be established and documented. Such validation and documentation may be accomplished in accordance with the 211 194A2. 21 CFR Part 211 Current Good Manufacturing Practices for Finished Pharmaceutical Product Support Laboratory. Method validation means established through the. What is the method validation? Just definition I will give you. Method validation are means established through documented evidence, a high degree of assurance that an analytical method will consistently yield results that accurately reflect the quality characteristic of the product tested. Okay, this is the definition of analytical method validation. So the objective of validation of analytical process is to demonstrate that it is suitable for its intended purpose. So ICS guideline for industry Q2A. In practice, it is usually possible to design the experiment work such that the appropriate validation characteristic can be considered simultaneously to provide a sound overall knowledge of the capabilities of the analytical procedure, for instance in specificity, linearity, range, accuracy and precision. So according to the ICH USP validation requirement, what parameter you have to perform during the validation activity? The first one is the specificity, linearity, range, accuracy, precision. In precision you have to perform the repeatability, intermediate precision, limit of detection, limit of quantification, robustness. So that's enough. It is the first part of the analytical method validation. Here we can discuss the what is the analytical method validation, in which regulatory agencies give the necessary guideline for the method validation, and what is the topic and what is the parameter covers during the method validation activity, and all the why the method validation is needed, what prior consideration have to taken before starting the method validation. All these things I will give you and discuss about things. So please, if you like the video and if you think something you get from it, please like and subscribe my channel. Thank you very much, and wait for the second part where I can discuss all the analytical method validation parameter individually and in brief manner. Thank you.